When we talk about the types of chemotherapy and CINV, generally we divide them up into three major groups. The first is acute nausea and vomiting. Generally that's defined as happening within the first 24 hours of chemotherapy. Then we talk about delayed CINV. That's generally considered to happen 24 hours to about 120 hours after chemotherapy. And then anticipatory nausea and vomiting. That's what happens prior to chemotherapy. It must be said that these definitions were first based on single-day chemotherapy regimens. We know today that a lot of regimens that are used are not only multi-agent, but they're also multi-day. So when we talk about multi-day therapy, sometimes these definitions blur. But for argument's sake, the major guidelines really define those three types of CINV in that manner. We know that acute nausea and vomiting, we do a much better job of controlling and preventing than we do with delayed nausea and vomiting. There are probably a number of factors and reasons for that. And certainly when we talk about delayed nausea and vomiting, that's probably one of the biggest areas of unmet need when we talk about this area of medicine and chemotherapy. We know that we do much better controlling and preventing acute nausea and vomiting. One of the reasons for that is we understand the pathophysiology, I think, much better than we do on the delayed side. We know that there's a number of neurotransmitters that are involved in this process. Certainly on the acute side, serotonin is the most important and kind of the major neurotransmitter that drives the whole antiemetic process within that first 24 hours. At some point, the neurotransmitters change. We think that substance P becomes a player in the delayed phase, and certainly dopamine is a neurotransmitter that is seen throughout the whole emetic process. We have agents that certainly do a good job of controlling the serotonin portion, and as I said, that's why we do a much better job on the acute side. We're still trying to understand a lot of the pathophysiology and delayed side, and certainly looking at now inhibiting multiple pathways in that delayed setting in order to better control a person's emesis and nausea post-chemotherapy. There's been a lot with patient education on CINV. For a long time, patients have expected to have emesis with their chemotherapy, whether that's due to television, and certainly the number of medical shows out there that show that as an expected part of their chemotherapy. So we're trying to educate patients that emesis with their chemotherapy is something actually that they should not expect, that having emesis has no way, shape, or form an impact on whether the chemotherapy is working or not. There are a number of tools out there that have been developed. Certainly we tend to use at our own institution the MAT, or the MASK anti-emesis tool, as well as the FLE scores, Functional Living Index of Emesis. These are validated tools that have been used in multiple clinical trials, case series, and most institutions will use them as part of their patient education and really monitoring of how well their patients are tolerating their chemotherapy from a emesis and nausea standpoint.